Hello, everyone. Thank you so much for the invitation. I mean, I co-organized, so it's not much of an invitation. It's really nice to be here and see many familiar faces that we had met either since last year in the first single-cell omics meeting in Stockholm, or from previous courses that we have been co-hosting together with the SIB and many other colleagues as well. So my flash talk for today is to identify training gaps in single-cell and spatial omics. So this is just a very broad topic, and many of the topics here also will be discussed later in the spatial part. But here is just a few ideas that we have been collecting in our training portfolio in Sweden, and we'd like to share that with you so that maybe we can actually collaborate a little bit on a deeper level. So one of the things of the training gaps that we actually miss is the pre-requirements for taking single-cell courses. Because in many cases, they need to understand a little bit what RNA-seq is, programming sometimes in Unix, or having a background experience in R or Python, depending on which course are you taking, especially because single-cell is already difficult per se. It's a very complex matter. So having the pre-requirements set, so having courses that actually give you a really good base for that is very important. So this is, for example, one of the courses that we have is RNA-seq data analysis that are together with Johan and Roy in Sweden. We have created also a full collection of videos about all the different kinds of analysis for RNA-seq. Yet, this is very far from complete. There are many other things that we would like to do, but we just don't have enough time to do all of this. So that's why having a more collaborative effort within all the nodes in Elixir and beyond, we might reach even a better state for not only for this course itself, but for all the others. So actually, this is bulk RNA sequencing, not single cell here, but this just a time series example how to do analysis on these samples. So this is bulk RNA sequencing. And this is one of the pre-requirements, or at least have a knowledge or have hands-on experience in RNA-seq to do a single-cell data analysis course. And this is also a course that is primarily based in R and Python using conda environments. Here we have some of the videos. So all of the lectures are also recorded, including special participation of Ahmed doing cell type prediction and labeling. But yes, there are many other things that I'll discuss in the few next slides that are still missing. And for this case here, so that's another thing that we could also potentially discuss, which are like the data set that we have been using. So far, we focus mostly on the single cell part, is a COVID PBMC data set, so from the blood. And they have patients versus controls and different disease status that you could compare. And so, yeah, it's quite broad and very like basic PBMC data set. Another one that we use and have been using like for 3 or 4 years now is the sagittal mouse brain for vision. So when you integrate single cell and do spatial deconvolution and just a very, this is a quite old data set now. So there are actually now data sets from all the different organs, and we could in theory actually have this matching single cell and vision data set and work throughout the whole week with them to get a better understanding and deep analysis, as well as for trajectory analysis. So in this case, we use bone marrow data set, progenitor of the immune cells are there. So we can kind of delineate the whole tree of cells. And these are the data sets used for that case. But I mean, we could use other different data sets for that. And that course is led by Åsa Björklund and myself, as well in Sweden. We have just one iteration in 2023, this January, actually. This course, we have set it up in different pipelines, so Surat and the bioconductor version, and also in ScanPy. So the students, they only work in Python, so they can just run the Python one, because all of those steps are exactly the same. It's just the code that is changing, but the concepts behind them are the same. So whatever pipeline you choose, you get the same result or very similar result, maybe different colors. There are still many, many issues. This year, for example, we had people now with plants, fungi, bacteria in single cell and want to do analysis. In some of our projects at MBIS, we had projects with krill and web spider granules, which I have no idea what it is. Some of those genomes are not even fully annotated, so they have single cell, and they are also doing annotation. So things are becoming a little bit more complex. It's not only mouse and humans. Although PBMC have been quite historically the gold standard for doing tutorials, Seurat itself only basically do PBMC, but there are more tissues that are require some more, maybe some data normalization, pre-processing that is a little bit different than PBMC, which is quite easy, let's say. And another topic is VDJ sequencing analysis. For two reasons, we still haven't included, although we have experience on it, but it's just a lot of work to create a course on it. And VDJ is already complicated by itself and requires also a little bit more of immunological background. So you kind of understand what you're actually doing, what VDJ are, and why are we doing this kind of analysis. So we would like to make a course. We don't have the hands-on capacity to build a whole course. If anyone has materials, and now I'm looking ahead with that all curated list of courses that you've published on LinkedIn, maybe we can find people to do that in a collaborative way. We've also had people working on single cell proteomics, so cytoph and flow cytometry analysis. But when they come with a course, they usually are aiming for single cell RNA-seq, but most of them already have other kinds of validation and wants to do single cell in these. Even though we actually have a few materials on that, it's not very much developed yet. So if anyone has a course focus, or at least part of it, that can handle this kind of data would be super interesting. We have also another course in attack-seq, but most of them still focus on these gene activity. So you just convert all the peaks to counts and do analysis, and that's basically standard single cell RNA sequencing. They don't usually go much deeper on that. Also, that it's something that we are kind of lacking in Sweden as training for that. And also for new spatial data sets, there are many different packages out there. One of the ones that I'm looking into trying, this is a package also just like SquidPy in Python for doing an analysis of spatial data in a much deeper level. And this one is in R. The single cell part, there is also some challenges in the spatial omics. So this is another course. The first iteration was last year, and it's a collaborative effort. As you can see, me and Navid were also here, also with folks from the Fabian Theis lab and also the other major universities in Sweden. That you can see the data generators for in situ sequencing, spatial transcriptomics, single cell, and interactive visualization tools for millions of data points with Carolina Belby. And in that course, we had only Python. We love, I do most of my analysis, the spatial part in R, but still to create a full course, everything in R takes a long time. So that's why I'm also looking for people that could collaborate and add to this, so we can have the Python and the R pipeline for analysis. And we had data sets with in situ sequencing, spatial transcriptomics as in there, and the single cell for matching samples. And all of those are already published. So we can do more these integrative analysis, gene imputation in situ sequencing data, cell type deconvolution using the spatial transcriptomics, or even a combined mixed model for all of them together. And we also have Codex, which is spatial proteomics, but so far we haven't touched much on that. One of the reasons is because it's a little bit underdeveloped, I could say. And that's actually, I'll talk that in my next slide. And all the videos and the lectures from this course is also available on YouTube. All in NV Sweden. I put also the link down below. So everything is open and accessible for all of those. We have many issues. So many tutorials, especially for data analysis in spatial omics, are either in Java or Python, which is not the most common languages. Python, yes, but not Java for data analysis. But also, so what we'd like to do is include them also in R. So just so we have the same parallel, same steps, you can do whatever with whichever language you'd like. And some of the tutorials were not linked for this first iteration. We try to put everything contained, so we can start and then you finish with something. But some of them are still a little bit hanging there. So we'd like to still polish the course. And this was just the first iteration. Spatial proteomics is still a field that I feel is a little bit lagging behind, especially in training materials. You have lots of them for the other omics. So like Merfish, Merscope, SMFish, any of the 300 spatial omics technologies that are there. But proteomics, many people still have these kind of like, oh, this is not RNA-seq, so I cannot use the RNA-seq tools, which has nothing to do one thing with another. But so I think one of the parts also would come from the course, meaning that, oh, if we show them how to do the analysis, then they kind of probably will start accepting that this is actually how you should do the analysis, not just say, it's protein, it's not RNA, then I should not do any single cell analysis on it. Spatial multimodal datasets are becoming available, but still, I don't know any good one to start doing courses on it. I have recently done also one spatial transcriptomics plus image mass cytometry, which is a good data set, but yeah, to do a whole train on that, we still haven't found a good one. A small advertise will have another iteration of, not of this course, but it's actually another course, Advanced Methods in Bioimage Analysis, which is the EMBL in Hardenberg, that will be here, led by Anna Clem from the Bioimage Informatics facility in Stockholm. And yeah, so this is an EMBL funded course, more on image analysis and so on and so forth. Some things that we kind of feel the need would be, for example, like maybe a data set glossary that we can find different data sets and why are they useful for specifically for teaching. So for example, in mind, I have maybe a data set from the small intestine, because you could do single cell, you could do trajectory because you have the stem cells from all the epithelial cell lineages. So it's a very clear trajectory, is easy to show. And we have space, and there are many sections on this right now. So it would be one data set that we could use to cover the whole single cell spatial transcriptomics and trajectory course that we have. Another thing that we probably need is a glossary with tutorials and parts. So for example, if we have a course on VDJ that we actually don't cover, at least we can point to that material. So for that specific topic, rather than referring to a whole course that sometimes have VDJ, at least we can try to focus on that. Any data set that has taxid collaborators here means any other omics other than RNA-seq, just because otherwise it doesn't fit putting all the names in here. So if you have any other data sets, a good experience how to actually work and make more sense of it other than just summing them to counts would be really nice and valuable also for us to learn. And another thing that we're planning to do is, as actually we have done that for the RNA-seq course, but we have this slide standardization. So that would be also great as a community if we start getting a little bit more on that, so that if we can actually reuse materials from other courses, at least they are already in similar format. So we can actually just, it feels at least for the student that this is part of a whole collaborative effort rather than things that were being scattered and put together. Data management tutorial, and now I'm looking at Amon. I've heard from Gabby, Amon is also joined. It's like, yeah, great. I'll see you again here. And I'll come to that because I'm also talking to Tony about this last week. Knowledge assessment tools. So for example, either before the course or during the course for reinforcement learning or as requirements. So for example, how can you check if the person actually has the knowledge or has done RNA-seq or have a little bit on R? at least a few questions so we can kind of assess that would be nice. I know, and that's another thing that I'm envious from the Finnish course, that they have, like after each lecture, they have the questionnaires like, oh, PCA, what did you learn about this? And like which statements are true, so that they can kind of like at least think a little bit after watching a video. And I think you probably will heard a little bit more on that later in the meeting. And yeah, so a collaborative effort is still what impedes at least us from Sweden also to get a little bit further, is like it's so much material that we kind of need to cover, and there are so many needs that we cannot do ourselves either. And yeah, so these are just some of the topics for just summarized from all the other slides that we can discuss.